The title of the project is QTL Editing for Crop Improvement, or QTL stands for Quantitative Trait Locus Editing. And the goal is essentially to apply the genome editing technology to this long-standing challenge of mobilizing agronomically important traits that are important for crop production, from one variety to the other. When plant breeders move traits from one variety to the other, they very often find that it's easiest to move traits that are determined by a particular single gene. And quantitative traits are a little bit more complex than that. They often are encoded, if you will, by genetic determinants at different locations across the genome, or their activity, how well they work, depends on the rest of the genome, sort of the genotypic background, you say, right? So it's really challenging when you do breeding to move those kinds of traits because they're complex genetically. And we think that genome editing will provide a real advantage, making that feasible and time and cost effective. There are three main areas in which we are applying the genome editing technology. Those three areas are: one, disease resistance, right? Two, tolerance to abiotic stresses. In this case, we're talking specifically about a plant's ability to grow well in acidic soil where toxic metals such as aluminum may accumulate. And three, what we call disease susceptibility. So it's kind of the flip side of disease resistance. There are genetic determinants that make a plant more or less susceptible to disease, right? And so we obviously want to find those determinants and move the ones that make a plant less susceptible to disease into our breeding material, right? So the objective of the project is not to develop new breeding material per se, but to sort of run the genome editing technology through the ropes with some well-characterized genetic determinants of these traits, right? To see how well it will work with these complex genetic changes that we need to make. Really, what I hope for this project is that we'll be able to demonstrate and to optimize approaches that will allow us and others to capture these traits of interest, these quantitative traits, and move them into different varieties so that basically it becomes a really powerful crop improvement tool.